as Dr. Streeter was saying, my name is Daphne, and I am a PhD candidate at Florida International University. More specifically, I am in the agroecology department. My main advisor is Dr. J. He's actually also in this presentation. Oh, there we go. Awesome. And so today I am going to be presenting on the effects of vermicompost on the growth and chemistry of medicinal plants. So just a quick overview of what I'm going to be talking about today. I'm actually going to be presenting a summary on my major projects of my dissertation work. I am getting ready to present present and also defend my dissertation. So I just wanted to give a summarized view of everything that I've been working on in the last 5 years. And so I'll also talk briefly about medicinal plants, a little bit about vermicompost and vermicompost tea, since I know a couple of my colleagues have already presented on this topic before me. And then after that, I'm going to offer some preliminary conclusions on my studies. And so I'll also talk a little bit of one of my publications, which is also relevant to the topic as well. So please, you could definitely interrupt me at any time if you have any questions, or just send the questions directly onto the chat and I could get to those after the presentation is done. So let's get started. So a medicinal plant contains compounds that can be used for therapeutic purposes or also drug synthesis. Observed pharmaceutical properties in plants can be attributed to the presence of secondary metabolites, such as phenolic compounds, terpenes, and also alkaloids and saponins. For those of us that are not super familiar with the term secondary metabolites, these are just non-essential plant compounds that play a role in stress, management, defense, and also sexual reproduction. So a good example of a secondary metabolite that some of us may be able to recognize might be like a smell from a flower. All of that are secondary metabolites that are trying to attract pollinators to a plant. So numerous drugs are derived from secondary metabolite production. A super popular drug that everybody pretty much consumes is aspirin, which is made from this plant called F. ulmaria. We also have morphine, which is something that a lot of people have used if they've been under surgery before. And this comes from a plant called P. somniferum. And so plant-derived medicine has actually been a source for healthcare for humans throughout history. In fact, about 85% of the global population uses some form of plant-derived medicines to treat disease or any kind of pain. And so as a result, the demand for medicinal plant productions is actually steadily increasing worldwide. The World Health Organization estimated that the global trade for medicinal plants is going to increase by $1 trillion by the year 2050. So an increase in demand for medicinal plant cultivation could lead to unsustainable production and unsustainable scaling up of this crop production as well. And so the cultivation of various medicinal plants is actually already associated with really high agrochemical inputs, which could lead to environmental degradation, also decrease secondary metabolism yield, because when you are fertilizing with inorganic fertilizer or when you're over fertilizing, that actually decreases secondary metabolite production in many plants. So for this reason, a lot of the farmers here in the United States are actually producing medicinal plants. They're actually transitioning into more organic agricultural management practices, and this could actually include the usage of vermicompost or any other vermicompost products as fertilizer. So vermicompost is a nutrient-rich fertilizer that's been created between the interactions of earthworms and bacteria. Vermicompost is a non-thermophilic process, and it's non-thermophilic because there's not the same amount of microbial activity as there would be in traditional compost. Something else that's really interesting with vermicompost that I always really like to mention is that it doesn't need to be a thermophilic process either to reduce pathogen load. The earthworm activity and also digestion actually controls the microbial populations that are going to be present within the end product or your vermicompost in this case. And there's been many studies showing that earthworm digestion actually significantly reduces pathogens within vermicompost, specifically coliform bacteria. So I thought that's always something really interesting to mention. So during the vermicomposting process, the breakdown of organic matter begins with earthworms. Earthworms are extremely important to this process because they drive this process by aerating, conditioning, fragmenting, or breaking down the organic substrate. At the same time, microorganisms present in the earthworm's gut and composting system biochemically degrade the organic matter by using enzymes or organic acids that they're actually exuding from their body. So vermicompost derived products such as vermicompost tea have actually become super popular in the last decade in agriculture, and this is due to its growth promoting abilities, and also it's just way easier to apply. When you use solid vermicompost, it's actually more of a slow release fertilizer as opposed to using vermicompost in a liquid form or in a tea form. It actually is an extraction of everything that is in vermicompost, so you'll have nutrients readily available for the plants to consume without any organic matter breakdown at all. So for those of us that don't really know what vermicompost tea this is, it's just a liquid fertilizer that's made by brewing water in vermicompost for 24 hours. If you choose to make your own vermicompost tea, sorry, remember that you do have to use non-chlorinated water or reverse osmosis water or pure water. If you use chlorine, you're actually going to kill the microbial activity within the vermicompost tea. So, oh, and something else that a lot of people don't think about is that you don't necessarily have to use an aerated tea. So when I produced my tea, I made it with an aquarium pump, but you definitely don't have to aerate it. That's just industry standard. Most people do aerate their teas, just to pump in oxygen and decrease the chances for it to become anoxic. And so beneficial effects of vermicompost applications on soil health and plant growth can actually be attributed directly to this biological activity and also to the presence of different plant growth promoting microorganisms such as nitrosomas, azospirillum, bacillus, and also different types of pseudomonas. Moreover, vermicompost also contains essential plant nutrients, phytohormones, non-hormonal plant growth regulators, also different organic acid exudates from microorganisms and enzymes that could stimulate change not only in the physiology of the plant, but also in the chemistry of the plant as well. So in previous studies, vermicompost has shown the capacity to increase essential oil and also pharmaceutical compound production in certain medicinal plants and also aromatic plants. So for instance, Kaur in 2022 observed an increase in total phenolics and also antioxidant potential in ashwagandha plants treated with vermicompost. Additionally, Caravas in 2018, he saw a significant increase in essential oil yield in two different mint species after vermicompost application. Mechanisms through which vermicompost and its byproducts influence growth and also phytochemistry in plants is not really well understood, but some studies do hypothesize that the improved nutrient availability and also the presence of beneficial microorganisms could be responsible for these changes that we observe in secondary metabolism during medicinal crop production. So something else that's really relevant to this specific thing that I just mentioned is that a lot of the times when you add vermicompost, exudates in microorganisms can also trigger defense mechanisms in plants, even though the plant is not actually being stressed. The plant does believe that it's being stressed, and that's why secondary metabolite production increases as well.
Number.
Yes, Professor. Let me see. Let me message her.
Yeah, or call her. Right? I think Jay is
calling. Yeah,
Yeah.
let's see.
Okay.
Yeah, Daphne. Yeah, I can hear you, but you dropped out of the the Zoom. Oh, no, no. Yeah, you dropped out, and we are like, I thought the internet went down, and then so we are waiting here.
Yeah, I might be.
Okay. So you were talking about I think that third or fourth slide, you were talking about different uh, the secondary metabolites and all those things, and then the aerated the vermicompost tea, and then yeah. We're almost about 7 minutes, like we are just waiting. And then we thought something. Okay, so you can yeah. Okay, yeah.
Uh, hello. I'm
Okay,
so
she's back.
sorry.
I
I'm
think,
so sorry. I didn't
okay.
even notice that the Zoom had dropped. I'm so embarrassed. I'm really sorry. Dr. Feeder, could you enable me to share again?
Yes,
Oh,
yeah, I made you a co-host again. Yeah, that's what I thought. Like, probably you'll keep going.
yeah, no, I did keep going and oh my God, I'm so sorry, guys. I'm so embarrassed. Oh my God. Okay. Well, at least I'm back, guys. I'm so sorry.
Yeah.
Was this the slide that I
Yeah, this the right. This the
okay.
one, yeah, almost,
Yes,
yeah,
yeah,
this,
this is
yeah,
the slide.
yeah,
Yes.
yeah.
Okay, I'm so sorry, guys. This very embarrassing. Okay,
No problem. Yeah, go ahead. This
okay,
happens.
so I'm just gonna repeat the slide, guys. I'm so sorry. I'm not sure where I dropped off in, but I will definitely be more alert about the I'm seeing. I'm so sorry. I'll be more alert about the Zoom. Since I am using two screens, I didn't notice since I was looking at my primary screen. In previous studies, vermicompost has shown the capacity to increase essential oil and pharmaceutical compound production in medicinal and also aromatic plants. So, for instance, Kauer in 2022, he did observe an increase in total phenolics and antioxidant potential in ashwagandha plants that were treated with vermicompost. Additionally, Caravas in 2018, he saw a significant increase of essential oil in two different mint species after vermicompost application. So, mechanisms through which vermicompost and its byproducts influence growth and phytochemistry in plants are actually not fully well understood. And so some studies actually hypothesize that they improve nutritional availability and also the presence of beneficial microbes could actually be responsible for the changes of secondary metabolisms that we have seen during medicinal crop production. So, to understand the effects that vermicompost tea applications have on the growth and chemistry of medicinal crops, me and my advisors actually developed two different studies involving turmeric and also basil. Can everybody still hear me? I'm like frozen in the Zoom. I want to make sure that everything is still okay.
Yes,
Everything
yeah.
good. Yeah, good.
Okay, sorry. Now I'm just really worried. So for those of us that are not super familiar with turmeric, turmeric is just a rhizomatous perennial crop that has been traditionally used to produce spices, dyes, and also different pharmaceutical products. Several studies have actually observed beneficial human health effects after turmeric consumption, and this is primarily due to its anti-inflammatory and also antioxidant properties. The medicinal properties of turmeric can actually be directly attributed to the presence of a secondary metabolite group known as curcuminoids. So, curcuminoids are yellow-colored polyphenols with potent antioxidant activity. They actually give turmeric that bright yellow or orange color that we see when we actually slice a rhizome in half. And you could also see it in the powder, like right here in the screen as well. Um, the major curcuminoid that's actually present in turmeric rhizomes is known as curcumin, and all the other curcuminoids are actually derived from curcumin itself. And so the other important curcuminoids that we can also find in nature and that we use a lot in pharmaceutical compound production is bis-dimethylcycurcumin curcumin and also dimethyl curcumin. So in the US, the demand for turmeric and turmeric products has actually skyrocketed over the last decade. The annual value of turmeric imports actually increased from $14 million all the way to $63 million. So despite high market value, only a few farms actually produce turmeric in the US and South Florida. And the production of turmeric in the US is actually primarily organic. And this is to produce and maintain high quality rhizome yields since rhizomes are very prone to accumulating heavy metals and also noxious chemicals from inorganic fertilization. Another reason why the USDA, which is the agricultural agency here at the United States, is pushing turmeric a lot is because it's actually a really high value crop, and they're trying to get farmers to grow it to be able to supplement their income, since a lot of farmers actually don't generate a lot of income from growing more popular crops such as corn or soybean. Another really big problem as to why the USDA is pushing turmeric here in the US is because we actually can't import turmeric a lot of the times as is. So we can't import turmeric in rhizomes. And this is because a lot of the times this will contain traces of soil and stuff like that, and that could bring in diseases and also different pests within the US. So a lot of the times we'll actually receive turmeric in powder form. And the problem with this could be is that other countries that might be growing this turmeric or other places that might be growing it may not be growing it organically, and that could actually cause really large amounts of concentrated heavy metals within the powder that you are actually directly consuming. So to pivot a little bit, I'm going to talk about basil since my second project is completely on basil. So basil is a subtropical culinary herb that's been grown worldwide as food flavoring and also preservatives. Although basil is primarily used as a spice, its essential oils can actually be used to make medicinal products for pain relief, gastrointestinal issues, and also oral health. Additionally, basil's essential oils have a strong antioxidant and antimicrobial properties, and this is due to a really high concentration in flavonoids and also phenolic compounds that are present in basil's leaf tissues, and some of these can be linol, eugenol, and also anthocyanin. So basil is actually super popular in the US, as I'm sure it is worldwide, since it's pretty much grown everywhere. And so for this reason, it's actually highly produced in many different states, including Florida, which is our home state, Hawaii, and also California. Basil can actually be currently purchased pretty much in any major retailer in the US. You could go to a hardware store like Home Depot and find basil. You could go to a grocery store like Walmart or also Trader Joe's, and you can also actually buy a basil plant as well. That's how popular it is here. And so additionally, basil essential oils and also supplements can be found in pharmacies and vitamin stores. A lot of the times, you'll only see a specific kind of basil, which is holy basil or Tulsi basil, but there is also other kinds of basil that are used for pharmaceutical compound production as well. So I'm going to talk about, again, as I mentioned before, I'm going to talk about my two major experiments for my dissertation, and these experiments explore different aspects of organic medicinal crop production and vermicompost usage in South Florida. The main goal of these projects was to understand the effects of vermicompost tea application on the growth and secondary metabolism of turmeric and also basil cultivation. Additionally, we really wanted to identify the mechanisms through which vermicompost could increase biomass production and altered phytochemical concentrations in medicinal crops. And I just wanted to show a background picture of our partner farm, which is known as Lion Fruit Farms. This is where I conducted the majority of my dissertation work. So for my first experiment that I'm going to be talking about, and again, I'm just going to give a summarized version of each one. So if you have any questions about anything, please let me know. The first one is titled The Effect of Vermicompost Tea and Compost Tea on Turmeric Yield and also Secondary Chemistry. So for this specific experiment, we did a really big field experiment at Lion Fruit Farms in Miami, Florida. We grew about 324 turmeric plants. We specifically had turmeric that was from the red Hawaiian variety, and we actually directly sowed them into the ground in large raised beds that were 3 meters wide and 56 meters long. We actually obtained all of the rhizome seeds from a nearby farm that we also partner with before, which is called L&B Farms. And all of these rhizomes were actually originally produced from 10 plants that he had gotten like years ago. So pretty much all of the plants that we were using were clones from each other. So there was low genetic diversity within the plants that I had chosen, and I purposefully chose plants that were clones from each other. So plants were arranged in a randomized block design, and a complete randomized block design, and each treatment was replicated 3 times to account for environmental variability, since we were growing outside in an organic farm, and there was trees and there was shading in some portions. So that's why we replicated it 3 times for each treatment. And so each block contained 36 plants as well. And you can kind of see it in this picture how the turmeric plants were arranged initially. So for treatment preparations, the treatments consisted of 2 types of liquid fertilizer. As I mentioned before, we did use compost tea and also vermicompost tea. And of course, we also had a control group as well, where we just added pure water instead of the liquid fertilizers. And so the liquid fertilizers or teas, these were prepared using local compost from Compost for Life Miami and also vermicompost from our partner farm, Lion Fruit Farms. And so both teas were produced with a 1:5 ratio of organic matter and also pure water. And again, we brewed this for 24 hours. So treatments were applied biweekly throughout a 7-month growing period. So that means that I actually technically fertilized my plants about 14 times. And each plant was actually treated with 200 milliliters of tea. And I was actually hand watering them. I hand watered every single plant and I directly poured it onto the base of the plant, right on top of where the rhizomes would be growing. So for this experiment, we collected a lot of different types of data, including physical measurements. We also collected soil nutrient content, chlorophyll production, and also curcuminoid concentrations in rhizomes. So for our physical measurements, we specifically collected aerial shoot height and rhizome yield. The reason I say aerial shoot height and not a stem height is because technically this like green part that you see on the top with all the leaves, that's actually not a real stem, that's a pseudo stem. Technically the stem for turmeric is actually the rhizomes. And so for our soil nutrients, we did analyze this through a Bruker XRF handheld analyzer, and also soil samples were sent to UF-IFAS USDA lab for analysis as well. Chlorophyll content was also monitored using a soil plant analysis development SPAD 502 chlorophyll meter, which I'll just refer to as SPAD after this since I do use it in the next experiment. And then finally, we chemically analyzed turmeric rhizomes by powdering all of our samples through a freeze dryer actually. And then we created an acetonitrile extraction, and then we actually ran all of the samples through HPLC UV-DAD to quantify the curcuminoids. So in this slide, I have a table with the physical measurements we took throughout our experiment, and the data is presented as the mean and standard deviation, and the letters next to the values represent significant differences within treatments or the control. So the results for this study demonstrate that compost and vermicompost tea are actually suitable fertilizers for turmeric growth. The addition of compost and vermicompost improved overall plant health and also promoted biomass production in general. So throughout our data analysis, we also observed that vermicompost tea treated plants actually performed the best out of all of the treatment groups, which is just compost and then also the control. And vermicompost tea treated plants had the tallest shoots. They had the greatest number of leaves, because I actually didn't include in this table. And also they had the heaviest rhizome yield, even though it wasn't significantly different. We also observed that the relative chlorophyll index or chlorophyll concentration was also affected by the treatments. The control group had a significantly lower chlorophyll concentration values as compared to the compost and vermicompost treatments. However, the compost and vermicompost treatments did not really produce different chlorophyll values. Actually, they were pretty much the same in the treated plants. So additionally, the soil nutrient concentration of 14 different elements was also analyzed at the end of the experiment. And there was actually no significant change in the nutrient concentration or heavy metals throughout the treatment blocks. However, we did observe that generally blocks under vermicompost tea treatments did have the greatest concentration of soil nutrients, including specifically phosphorus and potassium, which are plant essential nutrients. We also observed something else that I thought was really interesting. In both experiments, we actually saw this, which was that there was heavy metal concentrations observed higher in, but also in the compost tea treated soil as well. So the concentration of 3 major curcuminoids, including curcumin, bisdimethyl curcumin, and dimethyl curcumin, were quantified through HPLC UV-DAD analysis. And if you look in the screen right here, I have a chromatograph, and I also have the retention times that I use to be able to identify these compounds throughout all of my HPLC analysis. And so a MANOVA analysis was conducted to evaluate curcuminoid concentrations across treatment types. And so the results indicated that there was actually no significant chemical differences between the treatments. Despite no significant changes in chemical production, generally we did observe that compost-treated plants had the greatest concentrations of curcuminoids. Additionally, we did actually find some chemical differences throughout the experiment. The production of curcuminoids could have been affected by differences in block location, shading, and also the presence of large trees, because I did grow in an organic farm between different trees actually. And so we observed that generally blocks with smaller trees and less shading actually had the highest concentration of all curcuminoids, which could mean that light penetration was significantly affecting curcuminoid development. A lack of chemical differences could also be attributed to the crop variety or the variety that I chose to grow for this experiment. Turmeric rhizome seeds used for this experiment, as I mentioned before, were clones from each other. So in turn, this could reduce the genetic variability and also the chemical variability as well within the experiment. And so finally, we have determined that curcuminoid yield will most likely increase with rhizome biomass yield for this specific variety planted. So an indirect increase of curcuminoid yield can be slightly observed in compost and vermicompost treated plants, since the rhizomes on average were larger for the treated plants. So now I'm going to move on to my second experiment. My second experiment was titled "The Effects of Vermicompost Tea on Basil Growth, and Secondary Chemistry." So I did a similar project for this particular crop as well as the previous one. So for our second experiment, which involved basil, I grew 256 basil plants in 5-gallon pots, and this was again in the same location as before at Lion Fruit Farms, which is our partner farm. The basil plants were arranged in a randomized block design, and each experimental block contained 8 plants and was actually replicated 7 times. Again, I replicated it this many times because it is outside, and I did want to avoid any biases from either shading or just the position that the plants generally were. So plants were also spaced 16 inches from each other. Even though it may not look like that in the picture, they are spaced 16 inches from each other to decrease shading competition among the plants. And this picture that I have right here is exactly how all my plants were actually arranged. You can't see it very well, but there is little tags that would help me identify what treatment each one was, because I did actually have to hand water these plants when I was distributing the fertilizing treatments. So for this specific experiment, it's actually way larger than the data I'm going to present today. I'm going to only be discussing the results for one of the treatment groups, which was the vermicompost tea, and compare it to the control group, just to keep it more relevant for the topic that I was talking today. And so the vermicompost tea was produced exactly the same as the first experiment. It was brewed using a 1:5 ratio of organic matter and water, and brewed for 24 hours as well in these buckets that you see right there. And so additionally, the vermicompost was again also obtained from the same place, Lion Fruit Farms, and the treatments were actually applied weekly for this experiment, just because it was like a 6 to 7 week experiment. And I gave the plants 300 milliliters each. For the control, again, that one only received pure water instead. So for this experiment, we also collected a lot of different physical and chemical measurements. The physical parameters that we evaluated included height, above and below ground biomass, and also total yield. And when I say total yield, that just means the total yield of the entire experimental group. Also, like, I quantified all of the yield for the vermicompost tea treated plants, for instance. So soil nutrients and vermicompost samples were sent to UF-IFAS for analysis, and they're analyzing them through ICP and also spectrophotometry. Remote sensing tools such as the SPAD and also a Lycor LI 600 porometer fluorometer were also used to monitor stomatal conductance and also chlorophyll concentration across time and treatment type. And then finally, basal leaf tissue samples were also collected across time and treatment type, and they're currently being analyzed by GCMS to monitor phytochemical changes in volatile compounds. The reason that we chose to focus more on volatile compounds is because these are super important for basil's aromatic properties, which is why most people buy it, and also basil's medicinal properties as well. So in this slide, I have a table with all the physical parameters we measured throughout the experiment. The data, again, is presented exactly the same, just says mean with standard deviations, and the letters next to the values indicate differences in significance. However, there is no significant difference. Vermicompost tea treatments did not significantly affect any of the physical parameters we measured. However, we did observe a positive effect on plant height, biomass production, and also total yield. In fact, vermicompost tea treated plants actually produced the largest plants within this entire experiment, including the treatments that I chose to omit for this presentation. Vermicompost tea treated plant group actually produced the most basil also as well in general. They produced 1.4 kilograms more basil than the control group. So in this slide, I also have a table with the data gathered from remote sensing tools, such as the SPAD and the porometer, which I just mentioned. And so we observed that vermicompost tea treatments did not actually affect chlorophyll concentrations and stomatal conductance at all. However, we did observe an increase in chlorophyll production in vermicompost tea treated plants, but again, the same could be said for the control plants, which had slightly higher stomatal conductance. But when I grew these plants, the plants were actually highly stressed. So this means that the plants were actually dealing with their stressors pretty much the same. It was actually really hot. I actually grew these plants in April, but in April of last year, it was already 100 degrees during like the week that I planted, which was like, I think like April 16th or something like that. So furthermore, the addition of vermicompost tea did not significantly affect soil element concentrations either. Vermicompost tea treatments vermicompost tea treatments actually did affect soil elements. I'm so sorry. And so vermicompost tea treatments actually significantly decrease heavy metal concentrations in soil. Heavy metal concentrations in soil already generally decrease when you add organic matter. And I will say that there was organic matter in these fertilizers floating around because they were actually pretty dark, chunky, and concentrated. Additionally, the microbial communities present in vermicompost tea could have also produced organic acids and other substances that can chelate or form complexes with heavy metals, which would have reduced the presence of heavy metals or their bioavailability at the very least in the soil. And so to summarize all my findings from both experiments that I talked about, vermicompost can be an effective fertilizer for turmeric. Vermicompost treated turmeric plants did produce the heaviest rhizomes, the tallest plants with the most number of aerial shoots and leaves. However, vermicompost treatments did not really directly affect curcuminoid concentration. Curcuminoid yield was indirectly improved, however, with increased rhizome weight. So the more rhizome weight you have, the more curcuminoid yield you are going to have. And so in the future, I would actually like to test another aspect of the secondary chemistry in turmeric. I would like to focus more specifically on the volatile compound group known as turmerones, which are also used in pharmaceutical production. And these also come from turmeric rhizomes. And so additionally, the preliminary results for the basil experiment revealed that vermicompost had no significant effect on plant growth, yield, chlorophyll production, or stomatal conductance. This could just indicate that some crops may benefit more from vermicompost nutritional composition than others. Although we did not observe significant differences between the vermicompost tea treated basil and the control group, generally we did observe an increase in biomass growth, plant height, chlorophyll concentrations, and general yield as well. So interestingly, we did observe a significant decrease in soil heavy metals. And so specifically, I didn't mention it in the slide before, just so I had a little bit more content for this slide, but we actually saw a decrease in copper and zinc, which are heavy metals that could bioaccumulate in the soil and also in plant tissues. And so this was specifically in the vermicompost tea treated soil. And so this could be attributed to increased organic matter from the liquid fertilizers that we're adding, or perhaps, again, this could have been from the microbial activity originating from the vermicompost tea applications. And so finally, I still have to finish analyzing my basal tissue samples through GCMS so I can gain more clarity as to how vermicompost tea applications truly affect secondary metabolite production in basal production. So these are two of my publications that I have out right now, but I'm currently working on the publications for these two experiments that I just talked about. I've actually been working with Vermicompost a really long time alongside Dr. J. I've actually dedicated like 8 years of my entire academic career to learn about vermicompost and to study the effects of vermicompost in different crops. And so I just wanted to talk a little bit about my master's project because I think it's always super interesting to bring up. So during my master's project, my advisors and I, we developed a project monitoring the physiological changes of tomato plants after vermicompost tea applications. So during my experiment, I did find that the addition of vermicompost tea actually had a noticeable effect on biomass and also chlorophyll production in tomato plants. And through GCMS analysis, I was able to see that vermicompost also changed the chemistry in the tomato plant. Since I did not measure the fruit, I only measured leaf tissues at this point. And so it actually increased the production of several secondary metabolites such as terpenoids and also waxy cuticles. So interestingly, one of the compounds that was significantly affected by vermicompost applications was this compound known as beta-philandrin. This compound is actually responsible for that characteristic tomato aroma that you smell when you pass by a tomato plant. And it's actually a known neurotoxin for beetles as well. So I thought that that was really cool that the plant was actually increasing phytoalexins or defense molecule within its tissues with the addition of vermicompost. And so increased production of secondary metabolites such as defensive terpenoids and also waxy cuticles, which is basically the outermost layer of the leaf, could protect the plant from pests and other stressors. So vermicompost in general can actually decrease pests and disease through changes not only in the physical structures, but also in the chemical profile of the plant. And so these changes are also the changes that are occurring in medicinal plants, and that's why we're seeing differences in secondary metabolite production. And so before I finish my presentation, I just wanted to acknowledge my committee members, specifically my main advisor, Dr. J, which I've been working with for a really long time, and also the rest of my committee members, Dr. Salazar, Dr. Shea, Dr. Valverde, and Dr. Bott. I would also like to thank the agroecology staff, specifically Jordan, that helped me do all of my experiments. Also, all of the staff from Land Fruit Farms, Peter and Mary as well, who helped fund this research and graciously gave me a part of their farm to be able to complete all of these experiments, to the FIU graduate school for the DA fellowship and also DF fellowship, which actually helped me finish all of this work as well. And finally, I was also funded by the USDA and NIFA Hispanic Serving Institution grants. And so these are my references, guys. Oh, let me stop the share. So does anyone have any questions? I know I talked a lot and I talked a little bit faster just because of the whole mishap earlier. I'm so sorry. I really did not notice that the Zoom had like gone away. It must have been my internet.
We're open for questions for Daphne. I know in the turmeric experiment, I have a question. In the turmeric experiment, you told about vermicompost tea and the compost tea.
Yes.
What is the difference between those two?
Okay, so the difference between compost and vermicompost is just generally the way that it's produced. When I chose the compost that I chose, I specifically chose that kind of compost or that producer because they produce compost from manure too, which Peter originally used to produce vermicompost from manure as well, so they could actually be comparable. But the difference is the production. So compost goes through a thermophilic process due to intense decomposition and also bacteria or microbe activity. And so it heats up upwards of like 130 degrees, I think Celsius or Fahrenheit. And then that actually kills and removes a lot of pathogens and aids with the breakdown of organic matter, as opposed to vermicompost, which is a non-thermophilic process. So this means that most of the breakdown of the organic matter is solely done by the aerating, fragmentating and digestion of the earthworm. And then microbes do also assist by breaking down the substrate through enzymatic digestion. And the difference is, again, that the microbial communities are also directly controlled by the earthworm, as opposed to in compost, they're controlled by this thermophilic stage. All right. Is there any other question, guys? And you could also send it through the chat as well if you don't want to talk. That's totally fine with me as well.
And did you did you analyze the nutrient concentrations in the actual vermicompost tea and the compost tea?
Yes. So I just received the results for the vermicompost. I actually sent it a while ago, but it's taken like a month or two to actually analyze it. I froze them in the field I froze them in the field directly with dry ice. That's the word that I was looking for. And then I sent them for a nutrient analysis a couple of months ago. So I got the results like 2 days ago now. So I didn't have time to analyze the results and put them in this presentation, but I did analyze the vermicompost, and I also analyzed the compost and the teas as well.
Yeah, the reason I'm curious is because you haven't seen much difference. I'm curious to know, is there a significant, what is the concentrations of the actual macronutrients like nitrogen, phosphorus, potassium? Is it like very low, or how much it is?
Yeah, so from what I could see, the nitrogen was a little bit lower, but the phosphorus and potassium was like pretty high. I wouldn't say like extremely high, but enough to feed the plants. I think that some of the issues in my project could have been that maybe perhaps I wasn't adding enough of the tea. However, for my second experiment that I had different vermicompost tea mixtures, I did actually see significant differences, just not with the vermicompost tea alone, because I did add other organic amendments such as seaweed extract, and I also added hydrolyzed fish, which did cause actually significant differences in chlorophyll and in other aspects that I measured.
I have a question.
Yeah, sure, go ahead.
Okay. Yeah, thank you so much, Daphne, for the good presentation. I'm just making a follow-up of the question asked by Dr. Sridhar. Did you also maybe determine the leaf nutrient concentration? I am referring to the major nutrients NPK, because possibly these ones may have an effect on the secondary metabolites that we are trying to determine.
Yeah, so that's something that I did definitely want to analyze, but haven't been able to. I am limited by resources and also time, since I did all of the extractions and collected everything pretty much and with one other student, so I was limited by time. But there has been many studies showing that there is a difference in NPK concentration in leaves after vermicompost application. So that would definitely be a great to my project, and I hope that in the future, if I continue working with vermicompost and vermicompost tea, I could like do those kinds of analysis.
Thank you.
Thank you.
Also, if no one has question, I have one more. So, did you look at the pH concentration in the soils? What is the range of it?
Oh, sorry, the what?
The pH.
The pH constant, yes, I did look at the pH concentrations of the soil, and that's something that I definitely should have mentioned. So the soil was actually for both experiments, we obtained the soil from the same place actually, even though there were beds, they actually had like a soil mixture that they were producing there, and the pH was relatively high, I think, for turmeric and basil production. It was actually around 7.2 to 7.5 in most of my plots. So it was kind of high. But I did observe that the plots that I treated with vermicompost tea and also compost tea in general had lower pHs, and this was of course due to the presence of organic acids within vermicompost itself.
And the reason I'm asking is sometimes if the pH concentrations are high, I know some soils here are 7.9 or even
Yeah.
8.1. So even though you are applying the nutrients to the soil, probably the nutrients are not available to the plants. Only a fraction of it is available to the plants.
Yes, yeah. That's definitely something that could have happened 100% as well, since I was growing in like really large beds as well. So that was a little bit harder to control the pH for that one.
Any more last questions for Daphne? Good. Yeah. Thank you, Daphne. Thank you very much. I recorded this one and I will post